Professor Michael Parker, take one.
Mark.
Move. Marking. Or not. Or something's
Third
going
clap.
horribly wrong. Third clap. Thank you. Now, I think you're still on manual focus there, Steve. You must stay on manual focus.
Second. No, I'm on the auto.
It's on autofocus.
No, it's on.
It says manual focus on the lens.
Yeah, but it zooms here when I.
No, focus, not zoom.
Oh, the focus. Yeah, right. I'm focused on one.
Do you want to just hold that and keep it at manual?
I think so. I'll get Hansen. Thanks, buddy. Okay.
Felix, could you take that up?
Can do. I'm going to shut the door.
So I'm seeing the screen and it looks good to have the computer on. So if there's anything in there that looks
Scientific?
scientific,
Because that's my house.
you can see your house from here.
Yeah, so let me see where I'd find.
Shall we hold on? Cut that.
It's not something
Oh, embarrassing.
It's the simplest piece of equipment we have.
Oh, that reminds me. In my last interview,
Yeah.
TV interview, I used that, so that might be useful to.
Oh,
Which is an enzyme involved in cancer.
We've a long way to go before we understand that bit.
Mm.
And what's your fixation with Harrier jump jets?
That's a long story.
Some of them, just like, that's a Harrier. What's a Harrier doing?
Yeah, it's a long story, but and there's two different stories there. So one of them is one of my hobbies is belonging to a whiskey society, and you're a
Yep.
whiskey, yep.
Yep.
Whoops! Oh, I shouldn't have
It's
done
okay,
that.
That's right. You'll come back.
So that's my whiskey collection. Just there.
Oh my God! That you should never have showed me. Put it back. Put it back. Put it back.
No, show me again. Show
Not
me
for
again.
television. Yeah, sorry about knocking that.
Andrew, we've knocked the mic. Is it still good for you?
Just count.
1, 2. Well, if you count to 10.
1, 2, 3.
Okay.
Yep. So it turns out that there was a guy who used to be in media, I can't remember his name now, who's been going to the society, and his hobby is making models of military aircraft. And so he asked me at one event what one that meant a lot to me, and I think the Harrier is one of them. And so the background to that is I was in the UK for 5 years doing a PhD, and that was during the Falklands War, and the Harrier was a pretty important part of all that. The other one would have been the Vulcan. I would have chosen too.
Oh my God, the Vulcan. Huge. Huge. I was under one once. Yeah, it was like, I couldn't believe how big it was. Do you remember the famous quote from the BBC on Ark Royal, I think, when I can't say how many jets were involved in the sortie, but I counted them all out, and I counted them
Yeah, I
all
remember
back.
that. Yeah.
Two.
Yes.
And I think they lost about 6 on the Atlantic conveyor when it went down.
Yep.
Yeah.
Yep.
So yes, that was a fun time.
Oh, look, I think both the Vulcan and the Harrier were very famous because they were really the first of new technology.
It was phenomenal. I mean, you had the Mirages coming in, and essentially the Harrier just hit the brakes. Whoosh, boom.
Yep. Yep.
Yes, okay, we share two passions.
Mm.
Okay. Hi. Pleased to meet you. I'd like to start with, why did you select science? Or how did you begin?
How did I begin? Well, in
Now, from
terms
high
of
school, I mean, there must have been something that, okay, well, we've had many stories. Somebody said, well, the queue was shorter. The modern history queue was too long, so I jumped to another one. But I don't believe that's your story.
No, it's so I first got interested in science in high school. Just found it intellectually stimulating. But as I got near the end of high school and thinking about what am I going to do at university, I did get that feeling then that I wanted to be involved in the discovery of drugs. and wondering how I did that, had no idea. So decided to take up chemistry at university, which is one of the sciences you require for drug discovery.
Did you do chemistry in high school?
I did chemistry in high school. Unfortunately, I don't know whether it's changed very much these days, but when I did science at high school, it wasn't taught very well. It was all facts and figures you had to memorise, and it's not the way to really teach science. But nevertheless, I think chemistry and biology were the two that were my favourite sciences at high school.
So you did at least 2 strands or 3 strands of science in high school. So
I did some physics and chemistry and biology. So I think chemistry and physics I would have done to year 12.
So you made your choice really early. You must have found it very satisfying.
Yeah, just the general thought of wanting to be a scientist, but exactly how I was going to develop was something that I just didn't have enough knowledge to know which way to go.
Was there any catalyst to that, or was it just something you discovered of yourself?
I have a little theory about this, and I'll segue into one of the buildings we have at Bio 21 Institute, a building that was opened up a year ago, and it's named after a famous Australian scientist called Ruth Bishop. And she discovered a virus called rotavirus that was killing millions of children around the world. And she wanted to understand, from the disease point of view, what is causing that. And the fact that it was a virus was only discovered because she collaborated with a scientist at the University of Melbourne called Ian Holmes. And Ian Holmes had a way of visualising infectious substances, in this case a virus, using a special type of microscope. So I say all this because we have a little museum in this building, which I could show you later, and the family of Ian and Ruth gave some sort of mementos of their life to us, and in both cases they had a microscope as a child, and so did I. I had a microscope when I was in primary school. And so I just wonder how many scientists start off with a microscope when they're very young.
Was one of those cheap, an auntie gave, or your mum and dad, or was it just a...
It's, they were sort of functional sorts of microscopes. Not very high power or anything, but you could see better than with the naked eye, I guess.
That's great. Funny, you never know, if you give a kid something, you never know where it's going to end up.
But I think a microscope gives you a little bit of curiosity about the sort of natural sciences.
Well, it blew me out when I first looked at a drop of water. It was like, holy cow, really? I'm drinking that?
Yeah, look, I guess the other thinking about a bit more is one of the big events that would have impacted on me and people of my age was landing on the moon. And I'm sure that had a sort of a thing there about, but gee, that would be interesting.
Oh, same here.
Get involved in.
The optimism of 1968, 69, and we can do anything was very much written into our past. Speaking collectively with this. Yeah. Anything is possible. It's a bit depressing now, but still anything is possible. Well,
there are. I think today it's so easy to look at the negative things, particularly social media, that people get attention by dwelling on negatives rather than positives. But there's still a lot of positives going around. So one that is controversial at the moment is a certain rich billionaire who's creating a lot of chaos in the United States. But nevertheless, what he's done in terms of space travel is just revolutionary, and that's not something that NASA's been able to achieve. It's taken somebody of Elon Musk's capabilities to produce rockets that can reach the furthest parts of our solar system.
It's also a lot. I think it also blends into the history of research, doesn't it? You never know where research is going to take you. I mean, I think back to the space program, and somebody said, to me, what's the good research? Was it no space program on a very simple level, no microwave oven? And that's just the beginning of everything that came out of the general research that went into the space program. Would you agree or want to enlarge upon that?
Yeah. In fact, there's a paper, I haven't got to read it out today, which describes how a lot of our everyday inventions really have come from very fundamental, how does it work, curiosity sort of research. And I think it's... I think one way to reflect on this is a lot of the great discoveries are coming from academia rather from commercial companies. Commercial companies are very good at developing an idea that'll get into the marketplace, but aren't very good at coming up with those ideas because they can be quite crazy. A lot of them don't work. They can be quite expensive. It's not an area that the industry tends to get involved in.
I'm just going to stop you there because your computer went black on us.
Yeah.
That's the screen's over.
Oh, we're going to go now. Thanks, Ted.
That's
Didn't
right,
get through
yeah.
the questions.
That was it.
So, okay, you took yourself off to university and...
Well, at university, I went to Australian National University in Canberra. Interesting reason why I got to go to that university. I was in year 11 at high school in Sydney, in a high school called Narrabeen Boys High School at that stage, and the ANU had an early offer scheme, so you didn't actually have to worry about your results from your high school certificate. You could just apply in year 11, and I got early admission, so I was able to relax in year 12 and got into, my preference was chemistry. First year was just general sciences and then really developing into chemistry. Wanted to do biochemistry, but there's a conflict on when the subjects were taught, so I had to do chemistry only and went right through.
Did biochemistry exist then? Or?
Yes, biochemistry existed. So now I'm a biochemist, but I've never actually done a biochemistry course. So, which is an important point because one thing I did worry about in science is that whatever you decide on early, that you're stuck with that decision for the rest of your life, and that's not true. You can always change and follow passions as they develop, and that certainly happened with me. So when I got to third year university, specialised in chemistry, I knew the sorts of chemistry I enjoyed, but still had that idea of wanting to be involved in drug discovery. So for my honours degree, I chose to work in a biological area of chemistry, and in particular an area called structural biology. And that's what I have been for the rest of my life, as structural biologist within the field of biochemistry. So the structural biology, chemistry is all to do with atoms.
You got there without me asking. I'll bring him back to our chemists and move up from there.
Yeah. So
what is the history of chemistry? Okay, before we get it's all about atoms, but have to discover the atoms first. From the get-go, when did that all happen? When was?
Chemistry goes back to the beginning of time as human species developed. From simple things of seeing things on fires, of seeing things change, boiling of water, discovery of metals, being able to shape them, all that is science. I think the big breakthroughs were a few centuries ago, where people understood the very elemental rules of how you can get chemicals combining together to form new chemicals. So, if you look at everything we've got today, it's based on, materials are based on chemistry. in terms of biology, I think the discoveries that diseases or the human body are made up of biological molecules, and that's proteins and our genes, DNA, hormones, and so on. They were discovered over many, many years. And with chemistry in the say 19th century, when atoms were discovered as the very basic building blocks of chemical matter, that led to a real understanding of how you could actually use chemicals to make things that you really want.
I'd like to unpack that. Do you want a glass of water?
I've still got my coffee here, so.
Okay. With all of the throat clearing thing, it's our age, right? Okay, there's so much to unpack that I'm trying to make simple, but complex at the same. I understand the complexity through the simplicity. So atoms were discovered when, roughly? We'll track it down.
Oh gosh.
15th?
No. I'm struggling to remember the history of that, but yeah, look, the idea of atoms is something that developed over a period of time. So there would have been people suggesting that the basic building blocks of matter are made up of atoms, and then it was a Russian scientist, Mendeleev, who put together a special table called the periodic table, which looked at all the different types of atoms. We call them elements: iron, magnesium, oxygen, and so on, and was able to identify their chemistry of how the way they react with other atoms and so on. So that was quite some time ago. And then you come to the 20th century, where then the physicists were starting to look, well, what is inside atoms? So for a biologist, that's not something that I get involved in, but there's a whole stream of physics that are very focused on subatomic science. So in terms of atoms and biology, it's probably worth me going sideways here. And this is a very important story, this one. So I guess in the 1800s, late 1800s, scientists knew that materials were made up of atoms. But what they didn't know is, well, how the atoms actually are organised in a three-dimensional way. And is that important? So what you needed is to actually see, look at the material and see the atoms in the material. And so in the early 1900s, there was no such instrument to look at atoms in any sort of matter. The most powerful microscopes at that stage could see insects, could see an ant, but they couldn't see the atoms within an ant, for example. And so it took a special invention which had its origins in Australia. So the University of Adelaide were searching for a head of physics, the professor of physics, to head their department in the early 1900s, late 1800s, and they were able to recruit an English scientist called William Bragg, and so he was recruited to Adelaide, and he was married, and he had a child called Lawrence Bragg, and Lawrence grew up in Adelaide, and he actually got to university in Adelaide, and he specialised in physics. And as Bragg Senior, William Bragg became famous, England were very keen to recruit him back to the UK, which he did do. He went to Leeds, I think. So his son, Australian, came back, and he went to Cambridge to finish his undergraduate work. In the summer of, I think, 1913, he was very interested in some experiments coming out of Germany. So if we go back a little bit to late 1890s, early 1900s, there was a guy called Röntgen in Germany, and he is famous because he discovered X-rays. And one of the most famous scientific photographs around is when he X-rayed the hand of his wife, and you saw the big wedding ring and stuff, so you could see the bones. So there was other Germans who were using X-rays to look at materials in Germany, and were coming up with patterns, what we call X-ray diffraction patterns, when you shine X-rays at certain materials. But the problem is there was that was interesting, and each material had different patterns. But how did you?
I'm just going to stop your flow there. We went black again, so we get to go back, and you can do a take. We'll get a run through.
Yep.
We'll start back with William Bragg.
Yep. So William Bragg completed his undergraduate work at the University of Cambridge in the UK, and then went on to do a PhD. And then one summer he was sent a photograph, or an X-ray photograph, of what we call an X-ray diffraction pattern from some material. It was taken by German scientists based on the discovery of Röntgen of using X-rays to look at skeletons, and the puzzle then is okay, these patterns seem to be unique to the particular matter we're looking at, but how do you actually understand what's happening there? So it turns out the X-rays, the big advantage of X-rays, they interact with atoms, whereas light microscopes, what people were familiar then, what we'd be most familiar with today, they don't interact with atoms, they interact with objects, larger objects. So how do you convert the patterns that were being seen by the Germans into atomic structures? So Lawrence Bragg was thinking about this, and he came up with an equation, a fairly simple equation, mathematical equation, that would convert the diffraction pattern into an atomic structure. And his first one or two structures from that was table salt, sodium chloride, which gave a big surprise of how the sodium chlorides interact. And from then on, there's been about 40 Nobel Prizes in X-ray crystallography based on Bragg and Australian scientists on that original discovery. So it wasn't until the 1920s, 1930s, that chemists actually got, so first physics in structural biology, and then it went to the chemists. And they were really interested as, well, how do chemical reactions work? What do they look like? So one of the famous crystallographers, another Nobel Prize winner, Dorothy Hodgkin, was looking at vitamins. And she discovered the structures of vitamins, and that gave structures we just didn't understand how they would look like. And then she subsequently went on to get one of the structures, one of the first proteins, which was called insulin. And I think everyone understands that insulin is a very important hormone in the body. So she determined, using X-ray crystallography, so the process of X-ray crystallography is you get the protein of interest and you convert it into crystals. Then you shoot X-rays at the crystals, and then you can use Bragg's equation to get the three-dimensional structure.
That's kind of cool, isn't that? You're talking post-1913.
Yeah, so that's 1930s. Look, the big breakthrough in protein structures was in the 1950s. And in fact, the people involved in that were my supervisors when so subsequently to ANU, having done honours in structural biology, I went to University of Oxford to do a PhD, it's called a DPhil at Oxford, in X-ray crystallography. So that's where I learnt, and those people who were teaching me were the ones involved in the very first protein structures. So I certainly met Dorothy, but I also met the other people involved in the first structures, and the first structures were of proteins where we could get a lot of them. And so no surprise is that the first structures were of hemoglobin and myoglobin. Hemoglobin is the protein in our blood responsible for taking up oxygen when we breathe it in, and it transports it to another globin called myoglobin, which delivers oxygen to our cells. But because there's lots of blood, it's easy to extract the hemoglobin and myoglobin from blood and to purify it and to form crystals. So maybe too technical, but the question is, why do we need crystals? So what happens with crystals is the protein molecules, or the table salt, sodium chloride, they line up in a regular array. So they act like an amplifier. So if you shine X-rays at a single molecule and you get this scattering of the X-rays, it's too weak with one molecule. You need a lot of molecules aligned up in the same way. And that's why a crystal is so important. So, so we get these crystals and shine X-rays, and then we get these three-dimensional structures. And why are they important? That's why I'm involved in it to this very day, is that the three-dimensional structures can tell you how to make drugs. So an example with hemoglobin is there's a disease called sickle cell anemia, which is caused by a genetic mutant in hemoglobin. So some people, and quite a few millions around the world, suffer from this disease. And so from the 1970s, where we had these crystal structures of proteins, atom by atom in three-dimensional space, we could actually look at those and come up with molecules, small chemicals, that will bind, in this case, to hemoglobin to make it work better.
Just going to go back. What's the disease do?
If you look at under a light microscope, the blood cells, the red cells, are very deformed, so they don't actually work properly. So you don't actually get proper oxygen intake, and so you get all sorts of side effects from not getting oxygen delivered to your cells properly. I can give you an example of what we do. So this three-dimensional shape here, this object here, is the shape of a protein. Now, with this diagram, we don't see the individual atoms because that's quite complicated. Be tens of thousands of atoms. So what we do in this sort of representation show the shape of the protein. So this surface shows you what the shape would look like, but the size of this protein here is if you look at this size and put it in the center of the MCG, and then
Melbourne cricket ground,
and then shrunk it down to the size of a cell, and shrink this down the same amount, then you actually get the size of what this is in the human body. So this is millionths of a fraction of a millimeter. So they're very, very small.
Can you say that again, but use the Colosseum?
Using
the Colosseum.
The Colosseum.
Because you say MCG, we know the MCG. Everyone in Melbourne knows the MCG.
The world should know the Melbourne cricket ground.
The world should know the MCG.
We're about to get American gridiron there next year. And Taylor Swift was there last year.
Well,
Yes, there you
that's all
go.
we need to know.
But yes, a Colosseum. So
You do the same sentence. Well, I was going to start with, what the hell's a protein? Because we're having trouble with that.
yep. So a protein is a biological molecule. What that means is that it's a molecule found in biological organisms. So whether it's a bacteria, an amoeba, a fly, an animal, a fish, a human, we're all consists of we mainly consist of water and bacteria, but for the besides the water, we're made out of atoms, and because we're biological usually means it involves carbon. Therefore, we're full of protein molecules. We're also full of genes, the DNA, which is made up of atoms, and also metabolites in our body. So these are small molecules that do important work in our body. So vitamins would be an example. So some of these we can make, but quite often with vitamins we need to actually ingest them in our system. So that's why certain foods are very important to us. So how we get these materials? Well, we eat and we digest. And the very first part of digestion is a certain type of protein called a protease. That means a protein that breaks down other proteins. So when we eat food, we have proteases, which particularly in our stomach, which will break down molecules. So in terms of proteins, it breaks it down to amino acids, and those amino acids are then built up again to form new protein molecules. So a human has about 26,000 to 30,000 genes encoding proteins. Some people might have heard about the Human Genome Project in the early 2000s, and that project was to discover how many genes does the human body have. And we sort of know the answer now, a bit complicated, but effectively the answer is between 26 to 30,000 genes.
What was the impact of that? I'll go back to the Colosseum in a second. We're weaving our way, but that's okay.
Sorry, one second.
Yeah, okay.
So,
Look, I'd say the impact of the Human Genome Project is just ongoing. So it's one of those things where the big surprise of, well, here's the genes that make up our body. I guess one of the surprises there is that there's plants and insects that have got more genes than humans. And if that sort of puzzles people, I think that's because with evolution, genes have evolved to do very complicated things with proteins, so we don't need as many proteins as more primitive organisms. So what do the proteins do in the body?
Yes. What does it do? I mean, I heard a frog's got 44,000.
Yep.
So it's more basic, so it's got more.
Yeah, well, human proteins can be a lot cleverer. They can join up with other protein to form protein complexes that do more complicated things. So they're a lot more sophisticated. And so biochemistry is the science of understanding how proteins and genes work. So in our body, each cell has got a set of genes, and those genes, like the blueprint or computer code for the proteins that are going to be made. So there's special protein complexes in cells called ribosomes where they can convert the information from the gene to the protein. So what you need is the gene, which gives you the code for a particular protein. You need the amino acids because a protein is made up of amino acids and in the right sequence. And what we know is amino acids, as they join together, they fold into a three-dimensional shape like I'm showing here. So in terms of disease, What causes disease? Well, there's a whole variety of things that can cause disease, but one is genetic, is that can be mistakes in our genes. So there's constantly our genes are mutating, and there are systems in the body using proteins to try to stop that happening or throw those genes out or correct them. But unfortunately for some sort of diseases, that's an hereditary disease and not much you can do about it. But because genes are the basis for proteins, it's proteins which are the target for most drugs. So if a protein is not working, our job is to try to find molecules that will make the proteins either work better or actually to turn them off if they're doing too much of what they're meant to do.
That's about telomeres and stuff, doesn't it? Okay. I'm digesting. Steve, you're digesting?
Proteases.
Yeah, that's right. Lots of proteins. My genes are all lining up.
So, proteins, they're the building blocks of life, really, but there's the variety. It's so difficult. I mean, we'll need animation to go along with this to try and get the simplistic version. So, because it seems to me everything comes from protein. What you're looking for is to attack, support, turn on, turn off different proteins. Would that be? Would you say that?
Yep. So, so I guess in the going back to the historical context of structural biology, in the 60s we got our first proteins. The interesting thing at that stage is, although we knew proteins were made up of atoms, they were a part of a family of materials called polymers. And a lot of work was done on polymers. These are the plastics we have these days in the 1800s. And that work showed that polymers don't have any particular shape. They're like spaghetti. So a string of atoms repeating all the time to produce polymers or plastics or whatever. And so it's thought proteins, they're polymers and they're made up of atoms, so maybe they don't have any structure. So when the first structures came out, it was a big surprise. They actually do have three-dimensional structure. And then the amazing thing then is the first structures that came out adopted particular types of structures. So everyone thought, oh, that's what all proteins look like. And then subsequently, with some very famous chemists, another Nobel Prize winner, Linus Pauling, who predicted other types of biological structures. And they will actually start to be found as we got more X-ray structures of proteins. So it's all this variety. And so it turns out that we have a variety of proteins, but we have a variety of various different types of structures. The proteins are absolutely essential. They are the basic building blocks of our body. I've already said that you're looking at me. That's proteins in your eyes, the back of your eyes, that are converting that information to your brain, taking those images to the brain, converting that into something real. You're probably smelling things, which is a whole bunch of proteins in your nose, receptors that are taking up molecules in the air and taking that information to the brain through electrical signals and converting that into information. Hearing, there's proteins in the ear that will help convert that noise into something sensible. So I was talking about ears and sound and proteins in the ears that will send that information to the brain. Our skin is a certain type of protein, collagen. And then you get down to, as I've said previously, the air we breathe. There's hemoglobin molecules that will take up the oxygen, binding to an iron atom within inside the protein, and that will be transported to cells via myoglobin, and then the cells use that oxygen for their energy to make proteins, and so on.
Okay, so what's the timeline on this? You're talking the first ones in the 60s. along with the space program. Really. So the early days of the space program.
Yep.
It's not a long time for all this information to have come out. How is that? Do you want to discuss that? Because I find that astonishing in our lifetimes. Here we are, both kids probably watching the Man on the Moon when Neil Armstrong first got off, and we're infancy now to, well, we think sophistication today, but how sophisticated has it become?
Yeah, look, the field has become very sophisticated. So if you look at the timelines again, the very first protein structures took 10 years to do. When I did my PhD, DPhil, at Oxford, the typical length of time to solve a structure was about 3 years. Now, why 3 years compared with 10 years? Well, we learnt quite a bit, but it's technology that's really important. Again, as X-ray crystallography owes its discovery found into physicists, it was another set of physicists that actually made the big breakthrough for us. There is an interest in what makes up atoms, as I said previously, which is electrons, protons, neutrons, and then funny things make those up. And so you needed particle colliders. And so one type of particle collider is called a synchrotron. And what the synchrotron does is it can eject electrons. So one of the substituents of atoms,
You
and it
go
can.
back and start that again if you like. It's
Right.
You sort of
Okay. So one of the big breakthroughs for crystallography and structural biology was physicists who developed particle accelerators. And one type of particle accelerator is a synchrotron. These were built by physicists for physics experiments. And the way these things work is that they can eject a subatomic particle called the electron, which is what is the basis of electricity, and injects them into a ring, and there's magnets around the ring, and the magnets keep the electrons in an orbit. And the electrons are going at the speed of, or close to the speed of light. The analogy that we usually use to explain a synchrotron is when you're a kid and you have a bucket of water and you tie a rope to the bucket of water and you pick it up and you spin it around your head. The water doesn't fall out because there's a force that is going at a tangent to the orbit of the spinning bucket. It's the same with the electrons. So there has to be a force at a tangent to the electrons, and that force is, amongst other things, X-rays. So it's a source of very powerful X-rays. So when the chemists found out that the physicists had developed, millions and millions of pounds to develop these synchrotrons, that they had a high source of X-rays, intense source of X-rays, then the chemists said, "Can we please have a day and just see this will help us out?" And it did. It meant getting the data from. You could do it from smaller crystals. You could get data faster. You could get much higher resolution. You can see more atomic detail with a synchrotron. The next big breakthrough was from chemists, and I'll get back to this in a little while. But the problem is if you have a very intense source of X-rays hitting a crystal, it'll melt the crystal after not too long. So what do you do? So can we freeze the crystals? But you have to freeze them at a very, very low temperature. And it turns out that the temperature required to freeze crystals so you limit that X-ray damage was liquid nitrogen temperature. So mine's 180 degrees Celsius, so very low. So what we do these days is we freeze our crystals in liquid nitrogen. So then we were able to then solve protein structures within a year or so. So the two other big revolutions in structural biology is one is the original structures, as I said, were based on proteins you could actually purify because there was a lot of them, proteins from blood and so on, proteins from muscle. But how do you get to some of the really important proteins, which there's less of in the body. And that was in the 1980s with what's called the molecular biology revolution, where scientists in the States found a way of introducing human genes, for example, into a bacterium and telling the bacterium to go forth and multiply. That meant that you get a lot of bacteria making that human protein, for example, and then you could actually extract the human protein from the bacteria using basic biochemical techniques, and then you would end up with a lot of protein which you could then crystallize and determine their structures. So that was huge.
And this is what, mid-80s?
Mid-80s. And then I'd say, so this is part of my journey, is having got my PhD at Oxford, I was then recruited to an institute in Germany called the European Molecular Biology Laboratory. Interesting origin to this institute, called EMBL for short, is that it was created in the 1980s, 1990s. And the reason it was created is the birth of this molecular biology revolution, being able to make proteins in bacteria, for example. But in those days, there's a lot of worry. Is this dangerous?
Sorry, what are the inherent dangers?
Well, they didn't know. If you're producing a lot of genes and proteins that they normally don't make, would the bacteria escape, and would they be dangerous for some reason? And so they started off with a containment facility. So there's a huge European, with the founding of the MBL, there was a lot of interest from countries in Europe to house this. It's like the equivalent of CERN, the physics where they smash atoms to find what's inside atoms. So EMBL was the biology equivalent of, let's see what we can do with protein expression in bacteria and so on. Turns out it wasn't, generally wasn't dangerous, and there's all sorts of rules and regulations about what you can and can't express these days. The founding director of the EMBL was John Kendrew, who won the Nobel Prize for the first protein structure, myoglobin, in the 1960s. So a structural biologist was the founding director of that institute. So when I went there in the mid-1980s to early 1990s, another revolution was occurring in structural biology, which I think everyone can understand, and that's automation. So the way we collected our X-ray data was on photographic film, very slow. You had to develop it, and it was really messy, very slow. And you can probably know as a media person that there's probably a time where you were dealing with. That's right. So.
Maintain our jobs.
Well, they're gone now.
So detectors were developed to measure the X-rays, so they were really quick. And at EMBL, we were one of the testing grounds around the world for these new detectors, and so I was involved in that early testing of detectors. So that made it possible now to determine structures of proteins in months and weeks. And because it was quicker and exciting results were coming out of it, then that meant there was competition going on. Every university wanted its own crystallography group because we wanted to be part of the action. And so that led to the growth in the 80s and 90s and onwards of protein crystallography.
It became hip, didn't it?
It did, yes, and still is today. And so every...
They've gone black. Okay, we can start with you. You want to pause too?
I just want to reframe something. But I would like to ask too, just protein expression on what's that? There you go, I'm set.
Yeah.
Okay, just got to remember to keep clicking
No,
this.
just that protein
Okay.
expression, yeah. Because I'm on a different size now, so I just want to cover that again.
Yep. So what is protein expression? Well, protein expression is the steps of synthesize a gene that might be in the human body or other organisms and insert it into another organism, quite commonly bacteria, and encouraging the bacteria to multiply and therefore to express a lot of protein. So our first step in drug discovery is first we identify proteins and genes that might be important in, for example, cancer. And when we identify those, we look at the gene and express the protein or make the protein, and then we determine the three-dimensional atomic structure as the basis for developing molecules. Maybe I'll sort of segue a little bit, because it's topical, into a recent example of drug discovery which most people would know, and which my institute, Biotic Institute, was involved in, and that is the COVID virus.
That's always popular. It used to take so long. Oh, look, we can do it really quickly now. Why is that?
Yes. So there's a few things here. So with the COVID virus, obviously, I should be careful here, but I think it's obvious.
Look, if you say something, we'll give it to you and you can look back and go,
yeah,
no, I'll redo that, or you'll do a voiceover.
Yeah.
So feel free to speak frankly, and if you don't get it right, we'll fix it.
Well, it would appear that the virus originated in China. And the early discovery of the virus was made in China. And when Australia got its first case, within a month or so of the public announcement of the virus, our sister institute at the University of Melbourne, the Doherty Institute, were able to get the blood from the patient and tissue, and was able to isolate the virus. And the Doherty's expertise in able to understand how you multiply viruses. They had the techniques that they were able to find the magic to actually multiply, and then actually, because you need a lot of virus to examine it, and they were able to do that in the lab. So what they did is, I talked earlier about the fact that microscopy was so important to determining the virus in the diarrhea, the disease that Ruth Bishop was looking at.
Sorry, just did that. You lost me a bit. I'm sorry.
Yep. Okay. So a little bit.
Bad.
Yep. A little bit earlier, I talked about one of our buildings is named after Ruth Bishop, who was a researcher at the Royal Children's Hospital just up the road from us, and she was looking at a diarrhea that young children all around the world were getting, and it was quite lethal. Millions of children were dying. She looked at the tissue of babies that had passed away. She thought it was a bacteria that caused the disease. She couldn't find an obvious bacteria in the tissue she's looking at, and then she had an idea. Well, maybe it's a virus. In those days, the way of determining whether there's a virus, the gold standard method, was something called electron microscopy, which is another form of structural biology, which I'll get into. Instead of X-rays, electron microscopy uses electrons. So they're one of the subatomic particles that I've talked about with synchrotrons. So an intense source of electrons get aimed at a specimen, and use this electron microscope to actually visualize if there is a virus and what it looks like. I can show that to you later. So the Doherty knew that to actually characterize the virus, to see this is a new virus, so what does it look like? So they inactivated the virus of the Doherty. There's chemicals.
Sorry, they
inactivated, so it was no longer active. So there's chemicals you can add to the virus to kill it without destroying its structure. And then they came across to here, and they put it under our electron microscopes. And so the very first images, which I can show you later, of the coronavirus.
It's the siren, isn't it? Yeah. We all heard it coming, and we went, please don't. Yeah, I think there's a siren coming, but I was wrong.
Professor Market, I can take 3. Market.
Thanks, Felix.
I'm monitoring battery life from outside. So
And camera set.
Right, okay. I was talking about the coronavirus and that the Doherty found a way of making a large amount of the virus so it could be characterised as a step towards finding drugs to treat it or vaccines. And so they used a chemical to inactivate the virus, and they brought it across to the Biotic Institute. So the Doherty Institute, our sister institute, is only 5 minutes walk from here. And we put it under our electron microscopes, and we saw for the very first time in the world what coronavirus looked like. And we saw these spikes on the surface of the virus, and this popularly became the spike protein.
So it was here.
It was here, yes. And so more work was done and shown that the spike protein is the major protein on the surface of the coronavirus, and it was obviously a target to develop drugs. There are proteins inside the virus that have been used for drug discovery as well, but I'll just focus at the moment on the spike protein. Because of the urgency of trying to find drugs, scientists from around the world were involved in looking at this. My own lab was involved in looking at the spike protein. We continue to work on that protein. The first step is to get a three-dimensional structure. Now, it turns out with the spike protein, the rotten thing won't crystallise. We've got very bad crystals of it. And the reason probably why it doesn't form crystals is that it's floppy. So remember, a crystal, we want a crystal because all the protein molecules line up exactly the same way, shoot X-rays at it and act like an amplifier. If we can't get crystals, then we can't use that technique, so there needs to be another technique. And this is where we get to electron microscopy. And I'll just go to an aside here about electron microscopy. So I've talked about a career in structural biology, and there are 4 different techniques, major techniques in structural biology. One of them I've talked about a lot, which is X-ray crystallography, and still is a mainstream technique. And has been used a lot for coronavirus research, but not on the spike protein, which won't crystallise. The second one, oldest one, is called nuclear magnetic resonance spectroscopy. We do a bit of that, but it is not as powerful as crystallography, so it's not used as often. There are 2 other techniques, electron microscopy and artificial intelligence. Now, I'll talk about electron microscopy. Up until the 1990s, say, electron microscopy was very good at getting what we call low resolution views of molecules, a bit like this, but couldn't give you atomic detail. It wasn't, it just didn't give enough information to show atoms. So it's called blobology because all you could see is blobs. So they were still quite useful. But really for drug discovery and understanding how things work, you really want atoms. And so the big breakthrough in electron microscopy to convert it into a major technique for structural biology were 3 discoveries. And this will ring a bell with you. One of them I haven't talked about is about software improvements. And so one of the 3 discoverers of modern electron microscopy, biological electron microscopy, he developed mathematics. I talked about Bragg and X-ray. So this guy developed some of the mathematics required to actually get atomic views of molecules. Another guy actually worked at EMBL when I was there. And his role was very esoteric. It's talking about something you talked about earlier, of just very basic research. He was working on how do you freeze water. Now, you might say, well, you put it in a fridge, but what he was actually interested in is water. Biological samples need water. Us, we're 78% water. We need water. Proteins need water. So if we want to visualize proteins, and viruses, which are a bunch of proteins and genetic material. We need, and I've already said with X-rays, one of the big breakthroughs was freezing the crystals. One of the big breakthroughs of electron microscopy was actually freezing the specimens, because the electrons are really harsh. When you blast X-rays at material, they don't like it. They'll be destroyed within seconds. So how do you freeze? And so this guy worked out a way of freezing water without it destroying the biological material. And so part of that was using liquid ethane. So now we're actually getting down to close to absolute zero temperatures to be able to protect a material long enough to collect data from blasting it with electrons. And the third one will not be a surprise to you after what I've told you, is the other big discovery, which was done at the University of Cambridge. That's where Bragg made his discovery. Is that a guy called Richard Henderson knew that the big breakthrough had to be with detectors. So the detectors up until what he was using were very inefficient. And he was able to develop the physics behind the modern detector, where they were very efficient in taking every bit of electron information and converting it to a signal. So the three people involved in that, Jacques Duverger, Richard Henderson, and I can't remember the third scientist.
I'll look it up and you can see.
Yes. Won the Nobel Prize a few years ago for developing modern electron microscopy. So when the Doherty brought the protein to us, not only could we visualise it, but it did allow then people around the world, the Doherty showed how you could make a lot of virus and therefore a lot of spike protein. You could actually get the sequence of it, so you knew how to make it. So people around the world were making these spike protein and using cryo electron microscopy, cryo because it's frozen, and to actually get atomic structures of the spike protein.
Hold it. Take your black. Wait, it's a fine art here, Tim. Cough, Stephen.
So,
Just let it fade.
so that sort of prompts me to say something else. It reminds me that we're in a very special scientific precinct at the moment. We're in what's called the Parkville Biomedical Precinct. And it's one of the top 5 medical precincts in the whole world in terms of output and the quality of the science. Some of the major players that make the precinct, obviously the University of Melbourne, the Medical Research Institutes, I've talked about the Doherty Institute, Bite1 Institute, the Florey Institute, Walter and Eliza Hall Institute, but the other sort of organisations I haven't included so far is the hospitals. Just next to me is the Royal Women's Hospital, just up the road is the Royal Children's, and just behind the Women's is the Royal Melbourne Hospital, and across the road is the VCCC, Victorian Comprehensive Cancer Centre, and within that is an institute, the Peter Mac Institute for Cancer. And so we've got a rich interplay between the hospitals, the medical research institutes and the university, making this one of the top 5 precincts. So it's no surprise that we hear in the background every so often ambulances going by because we're in the hospital precinct, but also reminds us as one of our big roles in medical research institutes is make medical discoveries that impact on patients. So our patients are just next door to us. So it always reminds us that the real purpose of why we do what we do. One as aside, maybe a little bit technical, but I think it's important. So vaccines. So how do you make a vaccine?
We started writing with cowpox and the smallpox.
Right. Okay.
Is
So.
that the first?
You've got me.
One of the first.
Probably, yes. So the idea of vaccines is relatively straightforward. Is that if you've got a for example, a virus or maybe a bacteria that is causing a disease, if you can get a part of that virus or bacteria and put it into the human body, the human body's immune system will use its cells, B cells, T cells, and antibodies to mount an attack against the foreign element. So one of our Nobel Prize winners in Australia, Peter Doherty, that's what the Doherty is named for, he was very centrally involved in understanding how viruses interact with the human immune system. As an example, with coronavirus and the spike protein, it's obvious that you wanted to get parts of the spike protein, inject them into humans and see if they mount an antibody response. So the idea is if they did, then when coronavirus then got into the body, then those antibodies would already recognize the spike protein on the virus and then attack it and destroy it. So that's the idea of the vaccines. There's this remarkable new technology that was developed due to the COVID pandemic, and that is the mRNA technology, messenger RNA technology. So what I haven't said earlier is I said genes code proteins, but what I mean by that is we've got genes that are made up of DNA, but to actually make the proteins, there's another type of nucleic acid. So DNA is nucleic acid, Na, and it's called messenger ribonucleic acid, mRNA. So it's the in-between guy between DNA, genes, and proteins. So this mRNA was under development for some years, and people were worried about it. Is you're dealing with the genetic system of the body, is this going to be dangerous? You've got to be very careful. Along comes the pandemic, and then all of a sudden the drug authorities say it's worth the risk. Obviously, it goes through a lot of animal studies. It goes through lots of human safety studies. These all had to be accelerated. But this developed the first vaccines. So I think this story will be lost in history, unfortunately, but so I'll say it here, because structural biology was absolutely central to the mRNA technology. And the reason for that is the spike protein is what's called a fusion protein. And a lot of viruses have these fusion proteins on their surface. And what it means is that when the virus gets into the body, it wants to get inside your cells because it doesn't have all its own genetic material. It has some very simple material. It has a protease, one or two other things, but it really needs to get into your cell to get all the other things it needs. That's why sometimes viruses aren't considered a real organism, because it can't survive by itself. It needs to infect people and animals, or both, to get to cells to be able to reproduce. And the first step in that is the fusion protein recognises cells. So in this case, there's a protein in the human body called ACE2, and it recognises ACE2. And when it binds to that protein in the human body on the surface of a cell that's going to be infected, this fusion protein changes its shape and gets inside and draws this virus inside the cell. So it's the very first step in infection. And so that's why spike protein is so important. And trying to stop spike protein interacting with that ACE2 receptor molecule. And so the idea is with the mRNA technology was to make spike protein. So rather than the actual protein itself, we make the messenger RNA. So remember, it's the in-between between the gene and the protein. So we're going to introduce a foreign messenger RNA, which will encode for the spike protein. But there was a big problem. And the big problem was that the fusion proteins are very unstable because they've got to have two states. There's a state when it's on the virus, and then the state it's got to change to get inside the cell. And the problem is if you just made a vaccine based on spike, you may actually end up with antibodies recognising the wrong form of spike, the form that goes inside the cell, which would be useless. So this leads to, and this is all about science, it's all connected. We all rely on other discoveries, and it's so important that something I'm very proud of with the institutes I'm involved in is we don't sort of specialise in one sort of disease, we actually go across diseases. And so there's another virus that's being worked on. I used to work on it a long time ago called RSV, respiratory syncytial virus. And that's been in the news a fair bit the last couple of years because they've successfully made antibody drugs to it. So, and this is a killer for young kids, kills lots of kids, just like Ruth Bishop's diarrhea, rotavirus 1. The RSV causes, can be lethal, cause very bad sickness. So the big breakthrough there was, I don't know how far you want me to go with this, so you can stop me if I go too far with this.
You can do it you can
Yep.
do the long one and you can try and shorten
Yep.
it.
So look, there's two different aspects to this. But really, when I was working on the RSV fusion protein in the 1980s with a small biotech called Biota, we were dependent on a collaborator at the University of Queensland called Paul Young to actually make the RSV fusion protein for us to do structural biology. Paul had all sorts of problems, and unfortunately that collaboration ceased, but Paul didn't give up, and Paul just came up with a way of stabilising the RSV fusion protein by knitting onto it a bit of protein from the AIDS virus. And it stabilised the RSVF and did very well. So when coronavirus, the COVID came about, the University of Queensland with CSL, who've got their research division at Bio21, said, okay, we've learnt to how to stabilise the RSV fusion protein. Can we use the same trick with coronavirus? So therefore, you get your spike protein and you add on using genes this HIV stabilising protein.
Sorry, you went blank just a critical time. I saw the laptop guys, or the main screen last long enough to get to the end of the explanation.
Yeah, I wonder.
Can I cut that for just a moment?
Yeah, well, I think, yeah, sure.
Okay.
Maybe